Started here. It's my honor to host and welcome Matt Anderson back. He is a fun fact. He was an undergraduate here at UW, and I think both Nicole and I overlapped. We were here at the same time that Matt was here, but and he's now associate professor at The Ohio State University. I met Matt at SOCNUS many years ago, since we were both Eastern Band Cherokee, and so and I had been admiring his work from afar. Certainly, a lot of his work doing his indigenous genomics work that he's going to talk about tomorrow at Wimmer. So if you're interested in going to that, that's at noon tomorrow down at Wimmer. He's the things I kind of admire about Matt. He's a co-founder of Indigidata and also the co-director of SING. And if you don't know what those are, they are kind of summer programs for tribal students to learn about genomics and informatics. And Matt has gotten several awards and grants to support these things, and they're continuing on, which is awesome. And I really admire Matt for that. And I think it's just amazing work that he's been doing in that area. Just looking at his CV, he's done lots of, his research is outstanding, his teaching and his outreach. And he's probably one of the most inspiring people I know, and I'm really honored to have you here. So we're looking forward to, listening to your science talk today and then your talk tomorrow. So welcome, Matt. Thanks so much. So
I think my job is to keep expectations a little bit lower after that. So we'll see how well I can do. Let me turn on pointer just so that way it's easier for folks to be able to see that are on Zoom. All right, great. So what I'd like to tell you all today is a story really kind of at the center of genetics as a concept. This idea about how inheritance is passed down and how different systems of inheritance can be passed down over time, specifically through an organism that we focus on as kind of our main workhorse in the lab, Candida albicans. And so I know it doesn't require a ton of attention, but just as a quick reminder, fungi serve as kind of a really nice hallmark organism for understanding how genetics works, because you can understand fundamental aspects of cell biology using both forward and reverse genetics as principles to investigate those. You can understand how different aspects of inheritance work through plate cycling, through meiosis and propagation of genetic material over generations, but it also has important aspects to health. And in thinking about health, we often center it on the human, but I like to just kind of demonstrate that fungi serve key processes in agriculture, the environment, right, and human health. And so when dealing with trying to understand how the basis of inheritance operates, fungi can both serve like a very clear, fundamental academic kind of perspective, but also can serve a very important ecological and health perspective to that. And when you think about health, we often center that within humans. This graphic is doing exactly that, where you see these different colored bars, which are giving the global burden of different health detriments on the left. This is why I get to use a pointer. Sorry, I like to walk around, so it's hard for me not to walk over and physically point at the screen. Nobody can see me doing that if they're not here, though. So we have the contributions to different kind of health conditions within humans on the left, and you can see that some of the standard or kind of considered more innocuous aspects of that, we have individuals affected into the billions of people. And then we have more species-specific diseases indicated over here on the right. And within this list, you'll notice candidiasis kind of over here on the extreme right side. And so this is a specific area of candidiasis called oral candidiasis. So the fungal disease burden that exists in the globe is actually really high. So understanding both basic processes and its application to human health can be done simultaneously. And what I'm going to try and convince you next is that this is only becoming something that is even more pressing on us. So if you look at data from a single medical complex in Brazil, what you'll notice is the cases of major fungal infections is actually quite low. But then somewhere around the 80s into the 90s, this starts to absolutely skyrocket. And this isn't something about an ecological variant within the environment. This is really an outcome of applying medical techniques that involve immunosuppression to patient populations. And when you begin using immunosuppression to treat cancer, or you use them in transplant kind of scenarios, the incidence of these fungal infections begins to take off because you've removed a lot of the innate immune responses that keep them at bay. And if we look at candidiasis, if we look at these fungal infections over the last couple of years, just here in the United States, what I'd like you to notice here is that these gray bars indicate fungemia cases or mortality associated with fungal infections, and they're increasing over time. And then you can see these orange bars indicating the fungemia infections resulting in patient death that are associated with COVID. So since COVID has taken over, the effect of systemic fungal disease that results in patient death have taken off in really unexpected ways. So both within kind of a longer historical context, as well as an immediate context in what's going on in human health right here in the United States, as well as other countries across the world, systemic fungal infections is becoming a more and more pressing matter. And just for context, these bars are mortalities per month, not per year. So what organisms are being involved in these fungal diseases? These are considered kind of the big four. And what I'm showing you here is the data that we can get from the CDC that's most current. Candida, Aspergillus, Pneumocystis, and Coccidioides represent kind of the big four fungal pathogens within most countries and certainly within the United States. And while the number of total hospitalizations and number of mortality cases may seem fairly low, what I'd like to point out is the mortality rates associated with the systemic disease are absolutely set apart from any other kind of infection that we have health data for. Bacteremia typically results in about 2 to 3% mortality rate. Viremia is not that high canonically. Certainly COVID has changed those dynamics a little bit. But if you present with fungemia, systemic fungal disease in the clinic, you have a 50-50 chance of walking out of that hospital. Within those set of species, you'll notice the Candida species kind of exists at the top of that list with the most number of cases. Candida species are the third most common hospital-acquired infections. So not only number of hospitalizations, but the probability of picking up an infection in the hospital is also incredibly high. And the incidence rates of those infections have been increasing by 8% annually over the range that we have data for, which, yes, is a little backdated, but the recent data would suggest that these trends have continued. Within the Candida species, I'll point out Candida albicans. That's kind of, again, our workhorse species in the lab. And the reason is it is the most commonly isolated Candida species for causing these diseases among this group of organisms. So I'd like to argue from the start, the Candida species are underappreciated pathogens. We're going to talk a little bit about why some of that is, and it combines the ability to affect human health with the ability to understand basic genetic principles within fungal organisms. So why is Candida albicans such an issue here as a pathogen? And it's really because it's a commensal. It's a commensal that's found all over people, in your oral cavity, it's found in your gut, urinary tract, and it's found on your skin. So you're literally covered in Candida, which is why it's able to be such an important pathogen within the human context. If there becomes imbalance within the human, it's prepared its sites, different sites across your body. What's interesting about the fact that it can cover all these sites is in opposition to most organisms that live with us. It's dealing with a lot of different environmental conditions to be able to do this. Different pHs, salinity conditions, temperatures, being exposed on the outside of your skin versus the inside of your internal organs. Other microbes. Most microbial ranges are actually fairly tight based on aerobic conditions, temperature, etc. Candida is dealing with all these different microbes and it's able to persist within these different contexts, right? So when you lose some of the host barriers that keep Candida albicans at bay, it might be kind of expected then that not only is it going to be able to persist within these niches where it's normally found, but it's actually going to be able to disseminate. So it can cause mucosal infections at sites like the oral cavities, that oral candidiasis coming back, or vaginal candidiasis, yeast infections. It can also penetrate into deeper tissues and it can survive and spread systemically. So it's able to access the bloodstream and proliferate in those conditions, again, because of some of this plasticity associated with the fact that it's a commensal within these places already. So now that I've introduced why Candida albicans is worth studying as a system, let me give you a little bit more of the details about what does a system start us out with. So Candida albicans, it's a single-celled yeast, looks very innocuous, about a 5-micron cell. It's got a really nice genome if you're interested in doing genetics. The diploid genome, it's also very compact. It's only 14 million base pairs, but it has about 6,500 genes in it. And it has SNPs that differentiate each of the 2 homologs, which is going to be important when we get into things a little bit later. However, unlike most eukaryotes and most yeast organisms, there's no myeloid program that's been identified within the species, but it is highly amenable to genetic approaches, which means we can carry out most of the forward and reverse genetic tools that anyone else does within their model species. So we can study it pretty well. And it undergoes a lot of really interesting phenotypic changes. The one I'm highlighting here is a differentiated state between white cells, which are those yeast kind of round standard cell types that you're seeing, and what's called an opaque state. So this state is the mating competent state. We're going to spend a little bit of time wrapping back to that one today because I'm going to be talking to you about mating, right? These are not the only cell states that Candida is able to enter. And if you talk to folks that are working on other fungi, every time a new cell state comes out in Candida albicans, you get made fun of because we have so many cell states in Candida now that everyone just assumes that we're making things up. So because we can do a lot of really nice genetics with the system, a lot of people have focused on understanding how Candida albicans adapts. It has to deal with all these different physiological niches within the host, and it has to be able to persist within those environments, as well as transit into new environments. So if we think about Candida albicans adapting, canonically, we've thought about 3 different processes: point mutations, standard asexual evolution, loss of heterozygosity. Because it is diploid, you can replace one homolog with the other homolog, or changes in ploidy. So either gains and losses of single chromosomes or multiples, aneuploidy, or entire complements of chromosomes. And so a lot of work has gone into detailing these different mechanisms and their contributions towards adaptation in albicans. When we began my group, and even into my postdoc, we started asking a slightly different question. Are there other mechanisms beyond these standard asexual processes that can contribute to adaptation as well? And so one we started asking ourselves about is this alternative process called parasex, so parasexual reproduction, which has been described in a number of fungal organisms, including Candida albicans, but really hasn't been investigated at a sufficient level to tell us much about its contribution to adaptation. And so we're going to talk about kind of 2 pieces today. The majority of the talk is going to cover the dynamics of parasex and what parasex can do. I'm going to give you the punchline now. Parasex is better at producing genetic diversity than meiosis. I'm willing to argue that statement. Hopefully you'll see why I'm willing to argue that statement by the time this talk finishes. So this screwed up non-meiotic process that parasex is, that you'll hear more about, I think achieves its job just fine. And the second part is the implications of aneuploidy on cell physiology as a result of undergoing something like parasex. So I mentioned this word a lot. What is this thing called parasex? What is this hypothetical non-meiosis but produces a lot of genetic diversity system look like? Parasex begins with 2 cells that are of opposing mating types. If you've worked in yeast systems, it's going to sound very familiar. You can think of these as fundamentally sexes within chordates. There becomes a differentiation to that opaque state, that mating competent state, whereas the white state, that yeast state, is sterile. In that mating competent form, those 2 opaque cells of opposing mating types send out and respond to pheromones such that those can undergo fusion. So once they undergo both cytoplasmic and nuclear fusion, you now end up with a cell that contains all of the genetic information from both parental cells. You move from 2 diploids to a single tetraploid. That tetraploid can be propagated stably forever. It's perfectly happy on its own. However, you can also induce this process called parasex, which is a tetraploid cell undergoing a process called concerted chromosome loss because it just begin spitting out chromosomes randomly. As it does that, it's going to return to something that's going to approximate more of a diploid state. This kind of speaks to that aneuploidy formation I alluded to. And there's some evidence that there was a small frequency of recombination that potentially happens as a result of this process. So parasex now kind of looks like meiosis a little bit. You have mating, fusion of full DNA content, but then you have a disordered, not an ordered halving, but a disordered process of chromosome loss. We've only been able to do this in the lab. If you look at the population structure of Candida albicans, based on sequencing of approximately 300 strains by MLST typing, there are 17 clades, and there was no clear evidence of any parasexual reproduction or mating within the species complex. So if it can do this in the laboratory, why aren't we seeing evidence of it in natural populations taken from people? This study was done in 2005 before whole genome sequencing was widely accessible. So 2 studies have readdressed that question more recently, both in 2018. So this first one was a sequencing study of 183 different commensal isolates taken from the guts of otherwise perfectly healthy people. And there were 5 strains. These strains here that are blocked, this half blue, half green, and this block down here, half magenta and half orange, are 2 blocks where you see roughly 50-50 contributions from different k-means groups. So breaking out different strains based on the likelihood that those strains have a common genotype. You can see that there are 2 clear contribution k-means clusters to those strain genotypes, suggesting that there's evidence of recombination to produce those lineages. We published a study that same year where we were looking for evidence of some of the same things following up a study where we had done genome sequencing a couple years prior, and we saw evidence of recombination within our set. So 21 clinical isolates taken from active clinical infections, and we see 2 where, again, if we're using k-means and we're color coding each strain based on a representation of a common genotype for that group, 2 strains here kind of look like kaleidoscopes. There's a lot of different genetic contributions from different clade genotypes that exist within these 2 strains that do not match their clade assignments. This is all work being done by a technician at the time, Joshua Wang. So his work demonstrated, along with the Roper's paper by Christophe Donfer's lab, gave genetic evidence for recombination and potentially parasex in natural isolates. So we started to ask, if this is occurring in nature, we have our parasexual cycle. How well is parasex able to fill in the roles of meiosis, which are ploidy cycling, segregation? We can look at that through ploidy reduction and recombination. To do that, we took 2 isolates from our genome reference strain and mated them to each other and looked at the frequency of ploidy reduction and recombination. When we look at ploidy reduction, the overwhelming majority of parasexual products are not diploid. That's just this sliver here of about 1 to 2% of products. The vast majority are actually aneuploid, highly aneuploid in many cases. So chromosome reduction is not particularly efficient within this process. If we look to ask about recombination, we did selective genotyping, so restriction site-associated DNA sequencing for a number of these progeny. And if we compare them to the 2 parental genotypes here, you notice, again, isogenic strains were used for the matings. Genotypes are identical as far as we can see. But in the progeny, you can see these transitions, right? Indicated by these different arrows, suggestive of recombination. And if we try and map that out on a per strain basis for those that we sequenced, you get somewhere between 4 and maybe 15 recombinations per genome. If you're interested in meiosis, this is not great, right? That's not a lot of recombination. So although we could say that parasex retains some features of meiosis, doesn't seem to do a great job at retaining those features. When we spent some more time thinking about this, though, and thinking about the way we were using sequencing to understand recombination, we realized the experimental setup was actually a problem. we're mating two strains that are isogenic. It's easy for us to look at a recombination where we move from heterozygosity to homozygosity. Great, we can do that. But if there's a recombination that maintains heterozygosity, such as this example here, we'll never see it. So is the issue for why we're not seeing recombination during parasex not because it's not biologically present, but because we're not assaying it well? So we went back, redid these experiments. We used the same 21 isolates we had used to understand whether or not recombination was happening in clinical strains. But this time we took completely different strain backgrounds for our parents. Tried two different matings, two different clinical isolates to each other. We underwent parasex with these isolates. We mated them and then induced parasex. We then sequenced them again using dd-ras-seq, so we could sequence hundreds of isolates for a relatively low cost, and then looked at the parasexual dynamics. When we did that, we could go back and we can map contributions from each of the two parents in the progeny more accurately this time. And this is what we see. This is all work done by a graduate student in my lab, Bobby Fillinger. If we take two strains and we sequence and we map them back to the relative proportions of their genome that came from parent A, marked in purple, parent B, marked in orange, or that are heterozygous, one from each parent marked in green, we see a lot of diversity. What this doesn't tell us is it doesn't tell us the number of recombinations. It just means that genotypic contributions can be actually pretty heterogeneous within a mix of parasexual progeny. Keep in mind, this is from one mating product. So once you've made, you have the potential to produce all these different recombinant progeny as a result of that one mating. To look at recombination, what we decided to do was to break the genome up. We would start at the centromere, assuming that not a lot of recombination can occur across the centromere, because the centromere, generally speaking, if you have too much recombination there, you're going to end up with dicentric or acrocentric chromosomes, and that's not good. We removed all aneuploid chromosomes from any progeny we were going to investigate. Most of the progeny were diploid, so that was easy because we'd screen them by flow cytometry. And then we move across the chromosome, centromere to telomere, looking for transitions in markers that we could accurately map to parent A or parent B, which means they were usually homozygous. Parent 1 was AA, parent 2 would be BB. Now we know exactly which allele came from which parental genotype. When we do that, the recombination dynamics start to change a little bit. If we look at the number of recombination events per progeny, we now see some progeny have up to 100 recombinations. Better, respectable levels of recombination now. And we see that the fragment lengths pile up at around 10 kilobases. Anyone interested in taking a guess at what the distance between our markers were in the selected genotyping approach? If you're thinking 10 kilobases, you are correct. Okay, so now we're hitting up against another technical hurdle. What if the issue is we're not detecting enough recombinations or more recombinations because our markers aren't spaced in a way that allows us to see things? Fine. Well, what we can say now at least is parasexual recombination is more frequent. We did two matings, and I'm only showing you data for one because the other mating went very differently, which is really interesting. So I showed you the mating on the left because it's more dynamic, to be honest. The mating on the right, though, is a separate mating, two independent genotypes. The progeny are now overwhelmingly homozygous for one parent. Really interesting. There are still a number of recombinations that occurred within this mating for each progeny. I'm not going to show you the data for time, but it does indicate that parasexual outcomes vary between matings to different genetic backgrounds. So just because two genotypes come together doesn't mean the same thing is going to happen at the end. You can end up with one mating where you have larger levels of recombination and a really heterogeneous genome in most of the progeny that are produced, or you could end up in one like the other mating where you really seem to have dominance by one genotype and many fewer recombinations per progeny. If we look at the phenotypic heterogeneity between parasexual products of these matings, we chose these matings for a reason. We chose the mating on top because filamentation is very common in our genome reference strain, SC5004, but the 529L isolate does not filament under any in vitro condition. We chose the bottom mating because P6002 is highly resistant to antifungal drugs. So we wanted to begin mapping out traits, and that's why we chose these matings. If you look at the progeny diversity looks almost exactly like you expect based on the genotypic contributions, which makes sense, right? But it demonstrates that not only can parasects scramble the genomes, parasects can produce new phenotypes for selection after. So in our top mating where we had a lot of genetic heterogeneity, we have a lot of phenotypic diversity. In the bottom mating where it pretty much all looked like the genome reference strain, we see no antifungal drug resistance because our genome reference strain isn't resistant. So phenotypes track. But we wanted to return to this question about recombination frequency. So we whole genome sequenced 5 isolates. When we whole genome sequenced these 5 isolates, which had not been included in the original ddRAD-seq data set, we see a stark transition. Whereas before we were getting around 25, 50 recombinations, up to 100 recombinations in parasexual progeny by selective genotyping, all the progeny we whole genome sequenced are now showing roughly 450, 500 plus recombination events. That's a lot. Same schematic. We're only using markers that are homozygous in appearance that we can track inheritance from. No heterozygous markers are being used. So there's no possibility we're confusing inheritance here. You'll notice again, our recombinations are stacking up all the way to the left. This is because we've lost the ability of using homozygous markers to be able to distinguish the parents. It suggests that recombination may be even more frequent than we can capture here because we don't have any more homozygous SNPs between the two parents to pick up the recombination events at all. So the progeny genomes can contain hundreds of recombination events. We thought this is extraordinary. We did not expect this. We expected a couple dozen, maybe 100 recombinations per progeny, not hundreds. So we started to ask ourselves, how does this line up against meiosis? And the easiest way to compare it was by calculating centimorgan distances. So the distance in base pairs between two markers that are recombined at a certain frequency. And I don't want to belabor this point too much, but centimorgan distances are obviously different than physical distances, right? The actual number of base pairs doesn't always match up with the recombination distances. The beautiful part is centimorgan distances for a lot of meiotic species have already been calculated. So we could go in, find what those centimorgan distances were, and then compare it against what we got from our Candida albicans mating. This is what we see. So we plotted base pairs for centimorgan on the horizontal axis, vertical axis, and the genome size on the horizontal axis to kind of normalize things, right? And what you can see is that the meiotic species all fall within this window where the dashed lines are 2 standard deviations from all the data points for meiotic species. Candida albicans is down here. What this suggests is Candida albicans has a shorter centimorgan distance. It's recombining more frequently than any meiotic species where centimorgan distances have been calculated so far. So not only is parasexual recombination efficient, it exceeds meiosis. Obvious question that falls out of this is what's promoting recombination? In Candida albicans, we know that cryptic repetitive elements facilitate loss of heterozygosity and aneuploidy and chromosomal fusions and stuff like that. This is work from Anna Semecki's lab at the University of Minnesota. You can see really nicely that chromosomal elaborations align perfectly with these cryptic repeats that exist within the genome. So we asked Anna if she could take a look at our data set, get a better understanding of where our recombination events are coming from. So if you map recombinations against those repetitive elements that exist within the genome, a snapshot of a portion of the genome could look like this. So we're looking for overlap between the black marks and the magenta marks. And if we build mock data sets, just iterating recombinations randomly across the genome and asking if our actual data set has recombination sites more frequently associated with repeats than our mock data sets, we see closer distances between our recombinations and our data compared to computationally derived data sets where we just randomly scatter them across the genome. So parasexual recombination appears to be using repeats just like mitotic recombination is. So recombination occurs in clinical isolates. Now we've gone in vitro to show that recombination as a result of parasex is occurring very frequently. What we haven't done is we haven't connected this back out to the real world. We have a really nice data set to do that. So we took advantage of Christophe Onfer's clinical isolates here and assayed this group B and this group A. We can deconvolute their genomes because their contributions from the k-means group here and here for the group here at the bottom, and B is from this group and a group up top. So we can map basically parental genotypes to these recombinant segments A and B and ask ourselves, what is the recombination frequency? What I'm going to do is I'm going to show you a snapshot of the pileup. So this is just IGV reads on the genome here. The two parents are shown on the top. You can see in parent 1, it's all gray, matches the genome reference. That's our genome reference strain. And the bottom, you have our other clinical, or you have one of the other isolate backgrounds. Sorry, these are both not genome reference strains. In the bottom, you'll notice that there are 3 variant positions, here, here, and here. And if you map those back to the contributions from either parental genome, they're different. First, you're het, then you're home for one parent, and then you're back to het. This is an IGV snapshot. This is only about 100 base pair window. So if we're seeing recombination within strains like this, it actually matches pretty well with our data for this idea that parasexual recombination, even in clinical isolates in the natural world, are undergoing recombination at really high frequencies. Using this data, we can then go and do some of the same things we did before. We can ask, how many recombinations per strain? What is the average track length? And again, you're going to notice the track lengths pile up pretty high on the left side. We have the number of recombinations quantified for either the 2 strains in the A group or the 3 strains in the B group here. You'll notice that the 3 strains have roughly 7,000 even on that first bar. Their distribution in the genome is entirely random, so this is not a bias based on genomic locus. And if we pull these apart and do it on a per strain basis, each clinical isolate appears to have undergone between 2,000 and 5,000 recombination events following parasex in people. If you think about the implications within the phenotype diversity I showed you before, drug resistance, filamentation, which is a core process to its ability to cause disease. This means if parasex happens within a human, the progeny that produced as a result of that mating could have massive implications on the health of the human host. And if it's hospital-acquired infection, that human is already incredibly susceptible to fungal disease. Bloodstream infections with a 50% survival rate. Okay. One of the things we don't know yet is where do those matings happen, the recombinations. I'm blowing up one of those 2 groups, the A group, and we asked, how many recombinations do you have based on the branch lengths here? Some of these recombinations may happen after mating. Loss of heterozygosity can look like a recombination. If you're heterozygous, you LOH, and now you're homozygous for one parent, it's going to look like a parasexual recombination. It might not be. If we map out the number of recombinations per strain for each of these 2 groups based on the branch length, since their most common ancestor, that node here, we see no association. They're really just grouped out based on whether or not we're looking at A strains or B strains. So recombination, probably loss of heterozygosity, following the initial production of these groups, A and B, has contributed very little to the total number of combinations that we're assaying. Most of them have probably occurred in the common ancestor to those groups that then diverged asexually. So we think that recombination probably arose prior to divergence, parasexual recombination. Okay. So that's it for the first part. I'm going to try and give you a small vignette on the implications of aneuploidy. As I showed you at the beginning, we see lots and lots of aneuploids. Most of our parasexual progeny are actually aneuploid. They're not diploid. So who cares? Why does aneuploidy matter? So this is our data from those 2 matings. It mirrors the data that we published a couple of years ago. Again, 3-4% of the parasexual progeny are diploid or close to diploid based on flow cytometry, but most of them are not. If we look at a number of isolates that we've been using that are clinical isolates, interestingly enough, approximately a third of these isolates were aneuploid when we got them. So aneuploidy within clinical isolates, within natural human infections, is actually incredibly frequent. So this isn't based on some characteristic of those strains either. These represent very different clades based on phylogenetics, different geographic origins across the world as different sites. So there's no clear correlate between this idea of being aneuploid and being around in a human. We started to ask, is there something unique that aneuploidy confers? Because the aneuploid chromosomes in these isolates were oftentimes different. So is there something that aneuploidy helps you do as a Candida albicans strain in the human host? And so to set this experiment up, which is all done by a former technician of mine, Anna Mackey, who's now at Duke for a PhD program, we took our genome reference strain, induced parasex, and collected a bunch of isolates that were either disomic, including the parents, or contained trisomies for different chromosomes. We then grew up each of those strains and split them and performed RNA-seq, or saved half the aliquot for RNA-seq and half the aliquot for selected genotyping by dd-rad-seq. This would more closely align whatever genotype we saw with the gene expression for that genotype without any new chromosomal anomalies arising. This is our strain set. So we were able to get aneuploids for pretty much every single chromosome. The intensity of the red here indicates the number of isolates with that trisomy. We're only including trisomies here. There were some monosomies and there were some tetrasomies. We ruled those out. We decided to keep it a little bit more simple. But we have some both complex karyotypes as well as some simple ones. And the first thing we wanted to do once we had this was perform the RNA-seq and ask a very simple question. How well does gene expression correlate with copy number? And it correlates really well. This wasn't particularly surprising. This is true in yeast. It is not necessarily true in humans, but it's true in Cerevisiae. So we expected that gene expression may increase on chromosomes that are increased in copy number, where the DNA here is in black and gene expression is in red, where it's normalized to our diploid control. And we can quantify that here, showing there's a clear difference between trisomic and disomic chromosomes across these strains. So great, expression correlates with copy number. Let's start asking more interesting questions than some of these surface ideas. We asked, is there something in common that aneuploids express that diploids don't? So for that, we can't take the first aneuploids and just group them together. So we separated our RNA-seq data out by chromosome and then took each strain, removing those strains that were trisomic for that chromosome, and compared our diploids to aneuploids that were not aneuploid for that chromosome. And they just walked through each of the chromosomes. When we did that, we got 566 genes that are uniquely expressed in aneuploids compared to our diploid controls. What was interesting about that is the responses that we saw on this. A lot of work has been done in understanding how aneuploids behave. Angelica Lamon's group, Rong Li's group, has spent over a decade studying this, and they've done a really good job showing that things like the enfolded protein response is important for this. We see aspects of that. We see oxidative stress overrepresented in our gene list. We also see carbohydrate transports, basic growth. And we see some really interesting kind of Candida-more-centric responses, things like adhesion. So is there an aneuploid response in Candida? The answer is yes. And it seems to be directed not towards I'm upset as a cell. It seems to be focused in specific phenotypes, specific processes within the cell. Adhesion, oxidative stress, growth, and carbohydrate transport. Is this just because they have more DNA? So when we did this experiment, we also grew up tetraploids, the mating products between our two parental strains. And it's not. If we look at the genes that are in common between our aneuploid specific expression and our tetraploids compared to our diploids, there is significant overlap, but if you line them up looking at their expression, there's no correlation at all between gene expression and genes that are significantly enriched in differential expression in our aneuploids and tetraploids. And the processes are completely different. As I mentioned, in aneuploids, nutrient uptake, oxidative homeostasis, stress, etc. In our tetraploids, fundamental processes, tRNA processing, ribosomal processing, ribosome biogenesis, none of the same categories were there. So our aneuploid response is not caused by simply increased DNA. It's not just that the cell's dealing with more material. We asked, is there a correlate between our aneuploid response and a stress response? To do this, we went back to really old RNA-seq data. When we compared our aneuploid-specific expression to a bunch of different stress conditions, we didn't see a significant association with any of them. This seems to be unique. What's interesting about that is you'll notice, here's oxidative stress, here's nitrositive stress. Even though you would assume it would be more similar based on the fact that we're picking up like redox imbalance within the cell homeostasis. Our aneuploid response does not mimic those responses. What was also interesting is the aneuploid response is independent of how much excess DNA you have. We have a range of both complex karyotypes and simple. If you look at the gene expression for all of our aneuploids across each of the chromosomes, ordered from left to right with increasing DNA, gene expression doesn't look different as you move across each chromosome left to right. It looks the same. This aneuploid response doesn't seem to depend on how much imbalance you have, just the fact that you're imbalanced, which is really interesting. So at the end of all this, we kind of came to the conclusion that there's something unique here about the Candida albicans aneuploid response. And so we started to dig in. How does that change the cells? And we started with an easy one, growth. Because if anybody knows, putting cells in a plate reader is pretty darn easy. If we compare our diploids here in black to our aneuploids here marked in blue, you'll see that all of them grow slower. This is not surprising to us. We knew this is true in Candida albicans based on a paper from Judy Berman's lab last year, and it's commonly true in Cerevisiae and other aneuploid cells. If you're aneuploid, it takes longer for you to undergo cell division. So you have slower growth rates, and that's kind of quantified over here. Okay, growth was an easy one. Let's move over to some of the harder ones. What about this oxidative imbalance in the cell? To do that, we can use a chemical called H2DCFDA. So it's got a long name. The important thing for you all to know is that once it enters a cell, it undergoes a chemical reaction and it's trapped. It can't leave. So it doesn't diffuse. You can load the cells and wash out. If it encounters ROS, it gets converted into DCF, which then fluoresces. So if we incubate Candida albicans with this chemical, you can see fluorescence. Good, it's working. We can show that we can get a linear dynamic in which Candida albicans is typically expressing the outcome of incubation with H2DCFDA. So it's a good tool that we can use to measure the total amount of oxidative free radicals that exist within a Candida albicans cell at any one point. When we looked at our diploids and our aneuploids, this is what we saw. This is really interesting. We didn't expect this. Our diploids have higher levels of ROS than our aneuploids, even though ROS imbalance was increased in expression in our aneuploids. Bizarre. In fact, most of our aneuploids have expression or have the levels of ROS that are more similar to more quiescent cells. Diploids that have been incubated overnight, they're overnight saturated cultures, they're not growing as fast, they kind of depleted medium. They look like those. They're almost quiescent-like. So how could this be? Aneuploids have reduced ROS activity, but why? And so we came up with this hypothesis. We thought, okay, diploids have a certain level of ROS in them that they're constantly dealing with. But if they're exposed to free oxygen radicals from extracellular sources, they can bump the expression level of those enzymes to be able to deal with it. What if aneuploids are already maxing out the expression of all their ROS enzymes, which is why we're seeing less intracellular ROS when they're growing under like good, happy conditions. If that's true, if they're already maxed out, if we hit them with extracellular ROS, they may not be able to take it, and they're gonna die. So we ran that experiment using the standard growth plate assay. We took our aneuploids and our diploids, exposed them just for 30 minutes to either 40 millimolar or 80 millimolar hydrogen peroxide, not massive levels, and then spot plated them. So only 30 minutes. This is a single exposure, plate to rich media. lose the ROS. And what you can see is that a number of our aneuploids, that short burst is too much. Immediately kills them. Okay? So aneuploidy seems to have imbalanced ROS homeostasis within the cell that has altered their ability to survive following exposure to exogenous sources. Because we're dealing with a fungal pathogen, this could be a macrophage, could be a neutrophil, could be almost anything within your body. These make aneuploids fundamentally distinct phenotypically from our diploid counterparts. Okay. So what I've been able to tell you so far today is two stories that really revolve around parasex and about how parasex produces this massive level of genetic diversity. The parasexual recombination dynamics are ridiculous, outnumbering meiotic organisms by a bit. This produces a massive amount of genetic diversity that is complemented by a massive amount of aneuploidy that can form within parasexual products that produce genetic diversity that has consequences on transcriptional expression. As I showed you in the aneuploids, you get a unique transcriptional profile defining aneuploids that contributes unique phenotypic outputs on those cells. Our understanding of why this is important to Candida albicans is because it's dealing with all those different environmental conditions that it lives within the host. It has to be able to adapt. It has to be able to move across your body. And to do that, it has to be immensely malleable to overcoming whatever new environmental insult it deals with. So with that, I'll just acknowledge folks in my group that did the work. I really highlighted stuff from Bobby Fillinger, who's here, Anna Mackey, who's here, and Joshua Wang, who unfortunately is no longer in our lab. He's in his program at NYU, as well as our collaborators and funding. And thank you all for being here.
Any questions?
Yeah, that's fascinating talk. I agree. Just some naive, well, they're always naive for me. So. One question I had, a really neat idea, the idea that the aneuploids already were maxed out for expression of those ROS enzymes, and so couldn't, I mean, they were doing, you didn't see as much evidence of those species because they were doing everything they could to keep those down, right? So two questions. So something about, so is your hypothesis that something about them, they are dealing with higher levels of reactive oxygen species? They're already stressed to do that. They're already induced to do that. And they're succeeding under non-stress conditions. They literally cannot take anymore. Do you see evidence of certain enzymes being expressed at higher levels?
Yeah, so the unfolded protein response is something that's very common within aneuploids across any eukaryotic system, from humans to mouse to Cerevisiae. The oxidative stress that's usually associated with the unfolding protein response is metabolic. You're building and you're breaking down stuff. And you're building stuff because you're imbalanced in your chromosome. So you're producing more RNA and more protein that you don't need. We looked to see if there was compensation within stoichiometric protein complexes for factors of our aneuploid chromosomes, and there's none. So you are actively building things that have no use, so you just turn them over. So we think that that's where a lot of the oxidative stress is coming from, metabolically building things up and breaking things down. And the breaking things down, you're going to create these free oxygen radicals. So that's where the stress is coming from internally. That's where the increased ROS expression to be able to deal with those enzymes, those metabolic enzymes, originates. And yes, they're
There's
massive.
some limit, right? They can't. Like, what is it about them that's? Why can't they make more of those enzymes in response to?
Yeah, that is a good question. I don't have a good answer for you. We haven't tried doing something like evolving aneuploids, but being able to maintain selection for aneuploidy. That would be the trick. That might give us an idea as to whether or not the cell can either increase the ability to overexpress those enzymes to be able to deal with an environmental insult. We haven't done that. I think it's just because at that point in the cell ability to wire itself to be able to respond to things, it isn't ready for an insult. It also hasn't been grown in an insult, so it doesn't expect to have to necessarily see that.
I'll have to ponder that a little longer. Can I ask one more quick question? So about the slower growth rates of aneuploid, is there a specific phase of the cell cycle that's problematic, or is it basically every the stages that you might imagine being problematic, you know, S and M? Like, what is there any?
Yeah, in Canada, nobody's looked at that at a cell level, but the assumption is based on Cerevisiae and other organisms that it's M and S. That's really it.
Okay. Just thinking about your rates of recombination, you're trying to figure out how to scale that, you know, to an evolutionarily relevant scale of rates of recombination. You know, in Cerevisiae, he said, here's how many base pairs % organ or something like this. That's what happens during one meiotic event. But it seems to be super rare that they do meiosis at all.
Yeah.
Right, most of their cell division is very sexual. So do you have a sense in your system whether they're maybe doing more recombination events per parasex cycle, but fewer of them, or are they doing them a lot?
Yeah, you're asking an important question that nobody's been able to get a good answer for. There was a paper in 1999 or 2003, somewhere around the beginning of the 21st century, where Ken Nickerson, who's in Nebraska, put 2 antibiotic marked strains into a mouse with the assumption that if you get products that are doubly resistant, those are parasexual matings. He did get products that appear to be tetraploid and that were doubly resistant. He did not calculate frequencies from that, and nobody's gone back to those experiments, which we have been thinking about more and more now that we're kind of digging into this is happening a lot when it does happen, to get a better answer for you. But right now, that's all the data we have.
Very nice talk. I wonder, is there any evidence from your lab or the literature that might support the hypothesis of the aneuploidy specific gene regulation might come from chromosome level interactions?
Interesting idea. We haven't begun looking at that. What we've started to do, though, is we've started to look at what are the transcriptional regulators within the aneuploid specific response that may be associated with producing it. So we've been looking at more transcriptional regulation than we've been looking at chromosome dynamics, for example. But I think that's something we need to keep in mind. We're interested, there's about 6 or 7 transcription factors in our data set that we're now currently trying to manipulate to see if we can induce aneuploidy or we can stabilize aneuploidy to see exactly what the role is now. But thank you for the suggestion. We need to think about that.
That's cool.
Oh. Maybe I missed it. Obviously, I'm a space case today. But did you have some explanation for the very different recombination rates you saw out of those
matings?
crosses? Yeah.
So a rotation student, I had to start working on a project. One of them's drug resistant. So we thought maybe if we put drug on the plate to induce parasex, they would push it from being mostly our genome reference strain to our antifungal drug resistant strain. It didn't work, which is interesting. He's joined my lab. We're still following this up, but it's not his main project. So we are going to see if we can continue to skew things back the other way. If we can't, then it would suggest that there's some issue, not with one of the strains itself, because the strain obviously exists, but there's some process during mating and chromosome reduction that for whatever reason is preferentially retaining SC504.
Could you look at more crosses? I mean, I'm just curious what sort of range of distribution of rates that you see.
So that's an excellent question. I'm waiting for the reviewers to ask that. We're going to do more crosses. We have some more crosses in the mix, but the process of doing parasex is not the easiest in the world, which is why there's not a lot of folks that are interested in really focusing on it. So it's going to take us some time, but we're interested in doing it.
Okay.
Nicole asked my question.
Oh.
So I thought you were going to screen, with screen for mechanism,
Which mechanism?
the increased rate or the difference in rates of recombination.
We could. So as part of our 2019 paper, we built a strain that was marked on the same homolog with 4 different selectable markers. So we could look at exchange on that one chromosome between markers. We can use that strain as a workhorse. It's super finicky because if you get a loss of heterozygosity or recombination event just during standard mitotic growth, it screws up your markers. But we could use that as a mechanism. The issue is it's also only in one genetic background, so we'd have to reapply it. Honestly, I think sequencing is probably the easiest thing. And our DD-RAD-seq, we're sequencing a strain for like $1.50 a piece, so it's pretty cheap.
For online question.
Oh, yeah. Should look at it. I don't. You want to ask your question while you're at it? There's one back here too. Can you get it?
No, I can't.
Why don't you?
X out.
Yeah.
That would be
Ask
a
your
good
question.
I'm also curious about these different rates.
Yeah,
And I was wondering if parasites happen in homozygous mating or only in heterozygous mating, and if the mating type influences.
good question. So for those that aren't aware, homothalosism is the ability for MTL idiomorphs to mate with themselves. And it's most common in MTLA strains. We have not looked for recombination in homothalic matings. We absolutely could. We'd probably want to start with divergent genotypes that are both MTLA again, but there's no reason for us not to do it. Our assumption going into that experiment was that we would see the exact same dynamics that we're seeing here, because the process, the limitation there would be the mating itself, not the parasocial outcomes from the mating. So our expectations would be that we see the same thing, but we don't know yet.
Questions from that? All right, thank you.